Thank you all for coming to the SURF Final Symposium, where our 8 SURF fellows will go ahead and tell us what they have done this summer. We're all looking forward to hearing about all this wonderful work. It was a wide variety of projects, some of which were under the PICASC umbrella, the Pacific Islands Climate Adaptation Science Center, and some of them were under the Hawaii Sea Grant umbrella. So you will see kind of a difference in the topics that were covered in these projects. But all of them worked for 10 weeks and did some great research. And we got together a couple times to do some professional development. And then the rest of the time, they've been working with these fabulous mentors who have been willing to give their time and knowledge to these students who we hope will be fabulous researchers of the future. So let us get going. We are going to start with Sam Chu. Sam is a rising junior marine science major at UH Hilo and a Hawaii Sea Grant SURF fellow. He says that he is, quote, studying sustainability of marine ecosystems because where we live in a changing world where knowledge gives us power to adapt and move forward. So why don't you go ahead and start us off?
All right. Is it Fletcher?
Fletcher is.
One second. It was on. Hold on.
There you go. Does it work now?
There we go. All right. All right. Hello, everybody. My name is Sam. This is my presentation. We are diving into Aiken-Thurday, studying the life history of Hawaiian reef herbivores for sustainable fishery management and conservation. All right. So in June, I was placed with the Hawaii Cooperative Fishery Research Unit under the supervision of my mentor, Dr. Lillian Tuttle-Raz. Her unit is part of a nationwide program that links federal, state, and university partners. In Hawaii, the unit's primary cooperator is the Division of Aquatic Resources, DAR. And overall, the unit's goals are to conduct applied research and technical support for DAR, as well as provide student training in fisheries management. Thus, one of the research priorities for the unit in alignment with DAR was the statewide Holomua Marine Initiative, which focuses on monitoring and conserving Hawaiian reef herbivores, populations, and communities. And you might be wondering why reef herbivores and why they're so important. So these herbivores play a fundamental role in the reef, eating and controlling levels of macroalgae that would otherwise compete with corals. There are 4 key functional groups or trophic guilds, which would be the grazers, such as Manini and Pukuikui, the convict tang and the Achilles tang, shown on the bottom right. The scrapers, such as uhu, the parrotfish, we're not really showing those as much. Browsers, which include kala and other large Aiken-Thurday species, shown in the bottom left, the unicorn fish, and detritivores, such as kole, that golden-eyed sturgeon right there. Manini, Pukuikui, kole, and kala are all members of Aiken-Thurday, commonly known as sturgeonfish or tangs. And it's important because in Hawaii, they compose a very large proportion of the reef herbivore community. This leads into the research focus of my fellowship, which was, how can we improve the management and conservation of Hawaiian reef herbivores like sturgeonfish by studying different aspects of their life history? Now, let's quickly break down what a life history is. This is how a fish forages, ages, grows, and reproduces during its lifetime. This is foundational information for effectively managing fish populations and fisheries. Really important when describing a reef fish's life history, many have what's called a bipartite life cycle. This means that they have 2 main stages to their life, one pelagic and one demersal, well, benthic. The first stage starts as a fertilized egg traveling into the water column, developing, hatching into an early larvae, then a late larvae, and this goes on for about a couple of weeks to a couple of months, depending on species, before they then go through settlement, which is the process of settling onto the reef, becoming juvenile, then adult, before spawning and starting the cycle all over again. This summer, I was lucky enough to get to work with all of the stages early and late, more so just combined as larval, and then juvenile and adult stages. And I'll briefly describe some projects in adult and juvenile. In the adult and juvenile stages where I worked with them before focusing a lot on the early larval stages of these fish. Studying the adult with Richard Massey, one of the unit's research associates, I explored the basic life history of surgeonfish with a focus on 4 of DAR's priority species, this being kole, manini, umamale, and ma'i'i. Here you see me working on dissecting fish. From these dissections, we collected a number of samples, weight and length, otoliths, which are these stones at the base of the ears that you can count the rings on, kind of like a tree, to get the age and growth. We have the gonads, which are really important for determining reproductive patterns and behaviors, as well as we took some ciguatera samples for just general knowledge to understand where ciguatera is. Ciguatera is a toxin that is of concern for human consumption. To study the juvenile life history stage, I helped with the Pakui project. Some context, the Pakui, or Achilles tang, is a food fish and formerly popular aquarium species. DAR noticed that there was a decline in the species in West Hawaii. And so in 2022, they set a moratorium for the fishing of the species for 2 years. When they did that, DAR came to the unit and tasked the unit with helping the adaptive management process. And since 2022, the unit has done a lot of this project, a lot of dissections, a lot of this and that, but there is a specific gap or a puka that the unit is exploring right now, which is where are these juvenile pakui and what drives their distribution? What drives their distribution? To this end, the person who's really exploring that is graduate student Annie Larson. I helped a lot with creating the methodology for checking for these fish using this ROV. We use this ROV to survey over 50 sites across the windward and leeward sides of the island in search of these juvenile pakui. As part of her thesis, Annie will use the data that we are collecting still to then create detection occupancy models that will help predict what environments do we see where these juvenile pakui are found. Having these models, knowing where pakui are found, will then help us make decisions on where to protect them. I was going to show a video, but it seems like there's some problems with that. Here's a beautiful little still. And then studying early and adult, then that led to my independent research project, which was studying the early life history, the larval stages. Focusing on the early life history of surgeonfish larvae, the unit started a collaboration with Jeff Maleisen, and that was to help him identify larval fish specimens from his Blackwater diving photography database. Jeff Maleisen is a published naturalist, accomplished photographer, and he has over thousands of photos taken on these Blackwater charters of these larval fish with recorded metadata, a GPS location, all of the above. It's a phenomenal database. Quickly, I want to talk about what Blackwater dives are. There are these dives where divers go to areas where there's about 5,000 feet in depth, and they sit about 30 to 60 feet in the water column, using these bright lights to attract planktonic species. And they also get some other pelagic species and large predatory species. This style of diving originated in Kona, and this takes advantage of the diel vertical migration phenomenon, which is this epic process, this epic migration of trillions of planktonic species going from the deep, deep depths all the way to the surface at night to then forage for food before sinking down in the depths at the break of dawn. During these dives, Jeff takes a high-quality DSLR camera, and he gets these photographs like the one on the bottom right, which show this jellyfish and this leptocephalus larvae. Comparing traditional larval collection to Blackwater photography, traditionally, samples have been collected using tow nets or light traps, and we have a photo of a light trap in the bottom right, a little covered up with the line of the TV, but we get specimens that sometimes a little scrunched, sometimes a little damaged. Comparing that to Jeff's photos, we see some clear advantages to his in situ blackwater approach. This includes undamaged specimens. Generally, his photos have a higher image quality. And because he's doing these on commercial dive, well, not commercial, recreational dive charters, he has more consistent access to otherwise less accessible habitats than we would. Taking these photos, there was a lot of analysis that had to be done. First, I organized the files according to metadata. Each image had a lot of information to be recorded from it. I recorded the meristic data, that being the actual counts of the spines and the rays on each individual fish from each photo. I literally went to each photo and I counted spine by spine, ray by ray, to see how much they had for dorsal, anal fin, pectoral fin, pelvic fin, all of the above, covering the entire anatomy of the fish. With that recorded data, that meristic data, I was then able to identify them. I attempted to identify them down to the lowest taxonomic unit available, namely genus and then the species of the fish, even though it's a larvae and it's harder to tell at this stage. I did this using Dr. Bruce Mundy's larval identification guide. Quick note, Dr. Mundy is a retired scientist from NOAA, and he spent his life doing this work, and it's really impressive and phenomenal. From these thousands of photos of larval fish, all of these hundreds of dives, Jeff has recorded seeing about acanthurids on about 12% of all of his dives. And from that 12%, he has about 39 unique photos of acanthurids from 2017 to 2024. From these, I identified about 30 unique individuals. Breaking it down, it was interesting. We saw that there were 8 Bakui among these larval photos, 8 acanthurids that we can only identify down to genus, 6 Menini, and then a collection of Moorish idols and other fish of the acanthurid, well, family relative species. Going further, I took the time, like when in the month these fish were collected, these photos were collected, and I plotted them according to the lunar phase. So it's interesting, looking at the lunar patterns, we see an interesting spike around the 3rd quarter moon. This also sees a spike with Bakui. It's interesting, we don't have enough data though to continue to do a serious statistical analysis, but we are interested to see how this continues to build over time with more photos that are collected by Jeff. We also plotted these photos and when they were collected seasonally based off the month. We see a relative spike in March, half of which are Bakui, and we see a notable lull in July and then August. Oh, it's formatting's off a little bit. And what's interesting about this is that we see a lack of acanthurids in the summer, and that's also when we tend to see the highest levels of settlement onto reefs. So that may be something we just have to continue doing that. This kind of information is extremely relevant to us as it can suggest when these species are vulnerable and spawning. So that helps us know when to not fish for them. Unfortunately, we don't have enough sightings as before to do truly meaningful statistics, but we'll be excited to continue working with Jeff to see how these increase month by month and how they compare to other fish families that he possesses photographs of. Geospatially, I took a subset of the data from 2021, 2023, see where Jeff has been doing these dives out in Kona. We can see the sites where he's photographed acanthurids in red, the main dive sites in green. There's honestly no obvious pattern to where he sees these fish compared to his other dive locations, and they all seem to just be clustered relatively outside of Kona, which makes sense as that's where these dive charters are going. But with that, truly, I've only scratched the surface of Jeff's data set, and there's many more research projects to be conducted in the unit. This did raise a lot of very key questions. Why were acanthurids so relatively rare on these blackwater photography sessions? Why did he only get 39 photos? But also, why proportionally were there more Bakui in these blackwater dives? There's some next steps to be done, and that is to continue exploring this blackwater data set and to also continue the other ongoing projects within the unit. Oh, this is formatted different. Some key takeaways that I had is that the life history gives vital information to understand reef herbivores in these ecosystems. And then also with more information, we can then better improve the management and conservation of these herbivores. I'd like to give a big mahalo nui to the members of the unit, to all the organizations that helped bring me here today, help me conduct this research, to some of the people that I got to collaborate with that weren't part of the unit, and to the 2024 SURF cohort. Thank you.
We have a couple of minutes for questions. Does anybody in the audience have any? If anybody online has a question, you can put it in the chat and we can check it out. Yeah.
Okay. Yeah, is there a known?
Sure, he's the.
You gotta hold it here.
Here. You gotta hold it. Here.
Is there a bias that's known in terms of fish taxonomy towards blackwater lighting? Like some fish species or families are very attracted to it versus others, which might help to explain some of that data.
Yeah, that's a really interesting question. The question was, is there a bias with some of the behavior that might cause why some fish are attracted to the lighting versus others? Yeah, it's really interesting to actually, having spoken with Jeff about this, because we asked these questions ourselves. He thinks that with the kui, they're kind of cautious, but they're weird about it. We saw that with the kui that they like to come near the light, but not actually go into it. They like to just kind of like be a little scared on the outskirts of it, just trying to like be near it, but like not in it. The behavior is I don't know about literature behind it, but the behavior is definitely something to consider, and that might be a very important reason for why they're attracted, why are some are not. There's just more research to be done on that specifically. Any other questions?
Sure.
Great presentation, really interesting science. I loved all the photography, great pictures. I'm curious more generally, you know, after this experience, the SURF program, what are you going to take out of here as far as, you know, how's this altered what you want to do next in your education and maybe even looking ahead professionally? Have you had a chance to think about that yet?
I have thought about it a lot. It's an important question. The question was, how is this going to impact basically my future? How has this changed how I've thought about things with regards to where I want to go? The SURF program has shown me many things. This experience has really introduced me more to the fishery side of science, of marine biology and marine sciences. I want to continue working with the unit. We've kind of discussed this before. I think it'd be a phenomenal opportunity for me, other people. I want to learn more about larval sciences. I want to learn more about these fisheries, because through understanding it and through kind of the key takeaway that I have, through a better understanding of fishery sciences and the life history, we can then make better decisions on how to protect and conserve these species. Thanks. Any other questions?
Well, I think maybe we'll move on to Maggie. Oh, okay.
Sweet.
You pass your mic off to her while I introduce her. And is that you completed? Okay, let's move on then. So Maggie Chen is also a Hawaii Sea Grant SURF Fellow, and she's a rising sophomore in the Global Environmental Studies program here at UH Mānoa. She said she thinks that humans should take more responsibility for the damages we're causing to ecosystems with our anthropogenic impacts. You have the floor, Maggie. And you want the mic too, don't you?
Hello everyone, my name is Maggie Chen, and this summer I did my internship with NOAA at the Pacific Islands Fisheries Science Center in the Life History program. And for this presentation, I'll be focusing on the age and growth of Pristopomoides zonotus, also known as gindai. Before I get started though, I just want to give a shout out to all of the amazing mentors I've had to help me throughout this summer. Dr. Evil Shemel and Dr. Eileen Nally, my two co-mentors, and Dr. Kristen Dahl, Dr. Michael Kinney, Aaron Reed, and James Barlow, who are all part of the Life History program. So just for a little bit of background, the Pacific Islands Fisheries Science Center is responsible for a few different species, including some pelagics and some bottom fish. The fish that's circled on the screen is gindai, and it is a bottom fish snapper. So the way that the Life History program fits into overall fisheries management is that one of our responsibilities is to provide age data. And this is because we can compare fish ages to a few different variables like length, weight, sex, gonad data, and the location they were caught. And this allows us to get things like population and stock compositions, age at maturity, maximum lifespans, and mortality. And this is important because things like growth rates, maturity rates, and longevity vary a lot between different species. For example, species that sexually mature later in age can be at a higher risk of collapsing due to overfishing because too many individuals get removed before they're able to contribute to populations through reproduction. And so we need this information and data to be able to understand population dynamics so we can continue sustainably managing these fisheries. And these fisheries are important because they provide economic value, they supply us with food, and they're also in federally managed waters, so we're mandated to maintain them. And if we don't do so now, that just means in the future, we're gonna have to create a lot of regulations and limitations on these fisheries to deal with the effects of overfishing. So how do we get those age estimates? Well, we work with things called otoliths, which is what I spent majority of my summer working on. So otoliths are sometimes referred to as fish ear bones, and they're found just behind a fish's brain. And they provide sensory information for the fish, so things like movement, position, orientation, as well as sound information. And we work with otoliths because they can be used as a biological indicator of a fish's age. So I'll walk through the methodology of how we process these otoliths. So the first step is to actually remove the otoliths from the fish. So for gindai, we use a chisel to get into the fish's head and then remove the otolith with a pair of tweezers. And once we've obtained enough otolith samples from different fish, we go to a step called embedding, where we place the otoliths in a silicone mold and fill it with epoxy resin. And this basically just creates a block to hold the otoliths in place so we can section them. So we use these really precise low-speed saws to cut these really thin cross sections of otoliths. We typically aim for about 0.2 of a millimeter, which is about the thickness of like a cardstock paper. And once we have those sections, we lay them out on microscope slides and go through one last step called cover slipping, where we put a thin layer of casting resin onto the sections and put a glass cover slide on top. And this step is mostly to provide clarity for when we look at the sections under the microscope. And the last step is to actually age them. So kind of similar to the concept of tree rings, where if you cut a tree down, you'll find rings that you can count to get an age estimate. Otoliths work the exact same way. The theory is that these rings come from seasonal changes in growth rates. So when it's colder and there's less nutrients available, they have a slower daily growth rate, which creates these compacted, less opaque sections in their bone growth. And when it's warmer and there's more nutrients available, it creates these faster daily growth rates, which results in more opaque sections in their bone growth. And when you put these two things together, it creates this alternating ring-like pattern in their bone structure. And this is basically a picture showing what we would be looking at under the microscope, and the yellow dots represent how we would age this fish. So probably around 11 years old for this fish. Okay, so moving on to my results. For this preliminary data, we were able to obtain 235 samples from the Commonwealth of the Northern Mariana Islands, or CNMI. And these two graphs are just distributions showing the frequency of age and length of those 235 samples. For our growth curve model, we chose to go with the von Bertalanffy growth function because it's the most commonly used in fishery science. The equation is on the screen, and we have a few different parameters, including expected length at age or t, and L infinity, which is asymptotic average length, our k value, which is the Brody growth rate coefficient, and our t naught, which represents the age when the average length of the fish was 0. So for a k value, we got 0.25 per year, which again measures the exponential rate of approach to asymptotic size. Our L infinity, which is the average maximum size that these fish will grow to, is 37.2 centimeters. And our T naught, which is the estimated age when the average length of the samples was 0, is negative 1.52 years. This value is a little strange, and we typically want this value to be as close to 0 as possible, but we have a suspicion that this is because we have a gap in our data for our 0 to 2 year age range, or like our juveniles. And so there's not much data to anchor that side of the curve down, or it could also be that we're not accurately estimating our ages for our samples, because it can be really difficult to age these tropical fish because they don't experience that seasonal change that's represented in their bone growth. So it can be kind of difficult to read these. And just to reiterate, the reason we're doing this is, as of right now, there's a lot of uncertainty and variability in this fishery. For example, the number of participants in this fishery is declining. So the table on the top left kind of shows how over the past decade, we've gone from around 50 participants down to just 7 this past year. And this itself could be creating the illusion that this fishery is being overfished because there's less fish coming in, when in reality, there's just less fishermen going out to catch the fish. But with it appearing like it's overfished, it can result in things like really low catch limits. So for reference, Hawaii's bottom fish catch limit is 492,000 pounds, whereas CNMI's is only 82,000 pounds. And to put into perspective, this is what a typical fish market in CNMI looks like. They're selling fish on the side of the road out of coolers. So you can kind of imagine how there could be a lot of uncertainty and why it's hard to get reliable data or catch-based data in situations like this. So our intent is to use these biological processing methods to get more reliant and like data that's not so reliant on catch, which is easily influenced, to hopefully create more accurate stock and population assessments. And that leads me to my last slide of future plans or next steps. We're hoping to obtain more juvenile samples so we can anchor that side of the curve down and hopefully get more accurate values. We're also hoping to look at sex-specific growth because there are some data implying that it's possible males typically grow larger earlier than females. And we're also hoping to compare this data to the Mariana Islands as a whole, because this data is just from the Northern Marianas. So we're hoping to see if there's any differences. Yeah.
Okay, do we have any questions for Maggie?
Like you said, the number of people participating in the fishery was declining a lot in the last few years. Do you have any reason why that is happening?
I'm not too sure about that at the moment, but I could ask.
Just curious.
I think 5 to 30 years, that's a long time. I wasn't, aware of how long fish like that can live. Do you know about what age they become reproductively mature and so therefore start contributing to the population reproduction?
My mentor says about 3 years old.
So that's a long time for reproductive age.
Any other questions, or online, if there's anybody who has a question, feel free to unmute and ask it. No? Oh, we got one in the room.
I just have a comment. Great presentation, Maggie. I am Maggie friend to our professional. And I just put a little perspective on it. This fish can live to be about 60 years old in Australia. And we think it lives a little bit, younger in Hawaii. But these fish are considered unageable for like 20 years, and they haven't been done. As you saw, the list is very difficult. Those are some of the best photos to take in out of program, thanks to Maggie, and we really value her and her time. These fish don't have really nice solid growth patterns, so the next step is also like starvation and things like that. These fish are difficult to age. And
Tropical trees don't produce tree anchors either, so it's the warm, cold seasons in the temperate zone that give you good tree rings. So
Okay. All right. Well, then let's move on. Thanks, Maggie. All right. Next we have Andres. Oh, I'm going to butcher your last name, Andres. I'm sorry. Jojoa, is that right? Hohoa. I knew I was going to mess it up. Andres is a rising junior marine biology major, joining UH Mānoa this fall, and a PICASC Surf Fellow. Andres says that as a biologist, I witness the wonders of heaven on earth. Despite humanity's impact on many species, our planet still harbors marvelous and almost magical forms of life that sustain us and make this world a place worth living. A wonderful sentiment, and I appreciate it. So why don't you go ahead and give us your talk. Oh, yeah. You walk off.
Thank you. All right, so my project is about the social behavior and the cognitive variation among Indo-Pacific cleaner wrasses. My name is Andres Jojoa. My advisors, Dr. Jose Ricardo Paula and Dr. Elizabeth Maiden, and that's me in Kaneohebe. So the Hawaiian cleaner wrasse, the scientific name, laboratories Phytophagus. So on the left side, you can see this is the fish I worked the entire summer. It's very colorful in appearance, and that has a reason, because this fish cleans other fish. So they want to have this appearance so other fish can identify them. And they're known as cleaner fish, and also they have clients. So these are other fish that they look for them and they want to get clean. So on the right side, you can see one of the cleaning stations. Usually they have precise places where they clean stations. So the other fish, they memorize those spots and they always come to these places. And these fish that they come to the stations, they're known as clients. So these are some things that I came out in my mind about cleaner fish. So they have 3 different jobs. So they're known as the den fish of the sea because they go inside the fish mouth, and they clean inside their mouths, but they also clean around their bodies. They're also known as the therafish of the reef. They also get close to the fish and they massage them. So they help them to kind of like communicate with them, but also they relieve the stress. And they're also known as the police fish of the cleaning station. So there are some papers where it says that when the cleaner fish is present, a lot of the big fish, the predator fish, they behave better. They're less aggressive, which is very cool. Just imagine when you're driving in the freeway and you see a police car, you slow down. So a similar thing. So let's start to know more about this cleaner wrasse. So on the left side, you can see a giant fish, and there are 2 cleaner fish, very small. They're usually very small, like 10 or less centimeters. So they're cleaning this fish. So their behavior is cleaning other fish. So, so their food is parasites. On the right side, you can see one fish, and all the different type of parasites that grow around their bodies. So they go around their bodies and they eat that, the parasites. So that's food for the cleaner fish, and that's like a mutual benefit for the fish and the cleaner wrasse. Some other aspects about that. So the clients wait sometimes to get clean. So sometimes if the cleaner fish is cleaning other fish, they just stay on the side just waiting for their turn. And sometimes there are different fish waiting for that, which is really cool. On the right side, you can see some other behaviors that fish they have. So when they're getting clean, they pose. So you can see on the right side, there is this fish like vertical and with the mouth open. So you can see like a lot of different behaviors that fish you will never see if the cleaner fish are present. Like on the left side, this is a parrotfish, but you can see there is like something around the parrotfish. This is a mucus cocoon. They produce this during the night, which the cleaner wrasse they also do. And I think it's fascinating. Also, other fish, they do it. On the right side, we have this orange band fish, which normally you're gonna see it black, but when it's getting clean, or the cleaner wrasse is around them, they turn lighter. Their skin turn lighter. You can see it's like more gray. So it's like in a certain way, it's to tell the fish that they want to get clean, and also the cleaner wrasse, they can identify the parasites easier. These are other fascinating things about the cleaner fish. The sequential hermaphrodites. So that means that they're all born female, and at some point they turn males. So it's something very cool for me. Also, they dance. So to tell other fish that they're ready to clean. It's really cool to see that too. They also do the tactile stimulation, is the same like the massage that I was talking about, and they help stress relief other fish. The brain size is big compared to other fish. That means in a certain way, there are some studies that they're very intelligent compared to other fish. They have really good cognitive abilities, and they live 10 to 15 years. They have a really good effect in reef health, and they're found in the Indo-Pacific oceans. And they're also very selective on cleaning. All right, let's start about the project I work on. So on the left side, we have the phylogenetic tree about laboratories species. These are all the cleaning fish. So if you focus your attention on the purple branching, you see that all of them, they come from the same ancestor. And after that, they divide into two ancestors. One is known as the dishonest cleaners. That means that these fish, the top fish, they eat mucus on the fish. They like the mucus. The mucus is more delicious than the parasites, but the fish they don't like. So we say that when they eat this mucus, it's a dishonest cleaner. And on the left side, we have the other branch, the other ancestor, where we have the honest cleaners. So there are some studies about the dishonest cleaners, but we don't have that much about the honest. So we have 3 of them. The middle one, the Rubrolabiatus, there are some studies that they're honest. And I work on the right side, the Phytherophagus, which is the Hawaiian cleaner wrasse, which is uncovered, but I uncovered it. So I'm gonna have data from that, but we're still gonna have to work on the pectoralis, which is on the left side, to see if all the 3 are honest. So the purpose of this scientific project, how does the relationship between the behavior and the cognition in Hawaiian cleaner wrasse compared to the Indo-Pacific relatives? And also, are Hawaiian cleaner wrasse capable of solving strategic sophistication tasks, same as the blue streak cleaner wrasse? So the first experiment, we deploy different GoPro cameras attached to a weight where we see cleaning stations. This is a video, but I don't think it's playing. Okay, this is one of the cleaning stations. This is in Kanama Bay. And this is kind of like the data. You collect a lot of bunches of data. This is just one part. And when we analyzed the data, we came up with different behaviors about the cleaner wrasse. On purple, you can see the Hawaiian cleaner wrasse compared to other cleaner wrasse. You can also see that we put some red and green lines. That means that these two are dishonest and the other two are honest. And the middle one, we don't know, but we're, according to our data, according to our hypothesis, they should be honest as well. So here on the left side, we have the species clean. In the middle one, we have how many clients were clean. And on the right side, we have how much time they spent cleaning. According to the data, it's kind of like similar among all of them. Only the by color, they're kind of like they spend less time cleaning, they have less clients, and they spend less time. And there is some purpose why that's happening. And we're gonna jump around right now from those data, which is kind of similar to this one, which is like more like different. So let's come back about how the cleaning thing starts. So usually the cleaner wrats, they start dancing. So telling people, fish that they're ready to clean. After that, the clients, they approach. And after that, the cleaner wrats, they eat the parasites, so they clean them. But after that, certain fish, they cheat. That means that that jolt is produced. That means that they ate mucus from the fish. When that happens, usually the fish, they run away because they don't like that behavior. So you can see on the right side, the graph on the right side, you can see the first two, they have a lot of jolts. So they're not honest that much compared to the other two. They're more honest. And there is a paper that on the left side, there is a paper that there was a bleaching event called El Niño event in 2016. This reduced the population of the Dimidiatus fish, which is this one over here. So they reduced the population to 20%, which is a lot. So that means that there were less cleaning stations in Australia. That happened in Australia. So this fish, in a certain way, it was cheating. And because the clients, they didn't have another option to go to another cleaning station, they have to tolerate that behavior. So, but our findings in Hawaii, the Hawaiian cleaner rats that we have is very honest. So in the first experiment, we collect that data and it proves that it's honest, but we ran a second experiment to know if that is really happening. So our second experiment, we collected 10 different cleaner rats in Kaneohe Bay. And we brought it to our lab, which is in HIMB in Coconut Island. And we have them for 2 weeks for acclimation. And after that, we teach them to feed on plates. After that, we start the second experiment, which is the cooperativeness test. And we provide 2 different food sources. The first one is prawn, which is the circles. And the second one is flakes, which is the triangles. And so we want to know which food sources they prefer. And we identify that they prefer prawn instead of flakes. So on the phase 2, we want to teach them to feed first on the food that they don't like, and after that, they get the one that they like. It's kind of like having the same thing that happens in the wild. They should go first for the parasites, and after that, the mucus, because if they go first for the mucus, the fish is going to run away. But if they go for the parasites first, the fish is going to stay there. And after that, we jump into phase 3. This is where we answer the question: did cleaners learn the lesson? This is where we put 2 different sources. So this time we jump from like 14 different sources to 4, and this is like half and half. And the other one, when we were training them, it was like 2 versus 12, but now it's 2 and 2. And the answers and the results is like they didn't learn the lesson, what we teach them. So, which is in certain way is good news, because this is like comparing to our first experiment, it matches. So you can see the 2 cleaner rats, the first one that is in Australia, that they went over the bleaching event, and we have the Hawaiian one. So on the left side, we can see that the Australian fish is passing the test. So they're learning they're learning, and the Hawaiian one is not learning. So Hawaiian cleaner rats, they're not like they don't have to learn certain things because they are living in a really healthy environment, but the Australia one, because they went through something very hard, they have to survive, and they have to have very better cognitive cognition. So experiment one, I went through over this, but experiment one, the Hawaiian cleaner rice is very honest, not many jolts. Experiment two, they didn't pass the test, so their lack of cognitive adaptation. So the current data indicates that the Hawaiian cleaner rice are more honest and don't employ manipulation strategies. So I want to thank Dr. Rachel Lentz and the Pacific Islands Climate Adaptation Science Center and Sea Grants for the funding and for having all these amazing experiments. Mahalo, gracias, thank you, and obrigado. Dr. Elizabeth Maiden for having me in their lab, the Hawaiian Institute Marine Biology. My advisor, Dr. Jose Ricardo Paula, is an amazing, amazing person. And thank you for listening. And I wonder if you have any questions.
Do we have any questions? Oh,
Hello, can you hear me?
yes, we can.
Oh, great. Really interesting. I love cleaner fish, so I'm very excited to see your research, first of all. Second of all, I was hoping to, I guess, have you go through a little thought experiment, maybe, that you may not have the answer to, but I just was curious. So here in Hawaii, I think we just have the one cleaner species. We don't have any other labroides, at least, if I'm not mistaken.
Yes.
But in other parts of the Indo-Pacific where these fish are found, is it the case that there is often more than one labroides and more than one cleaner in the community? And I was just curious what your thoughts were about how the impact of having, I guess, if the cleaner has to compete with other cleaners of different species for clients, do you think that that might impact their cooperativeness or their willingness to cheat?
Yeah, I don't have the answer for other cleaner rice around the world, but I have the one in Hawaii. We only have one cleaner rice in Hawaii, but we have multiple of them, and they have their certain spots, and sometimes they come. I have seen three in one spot, but I don't know if they're coming from the same community, or they grew up together. But I just imagine how that would happen if we have multiple of them at the same spot, and like different clients might be present there. So I think if there are two different species in the same spot, and I think they might have a better behavior, so they're gonna be more honest because they don't want to lose their clients. So that's what I'm thinking about that, but I'm not sure.
Thanks. Yeah.
Yeah.
Okay, I think we're going to have to move on. Thank
Yeah,
you.
thank
That
you.
was great. All right, we're going to shift from fish to corals. So we're going to move on to Maria Plapral, who is also a PICASC Surf Fellow, is a rising junior at UH Mānoa, studying marine science and data science. Put this thing up here. There we go. Oops. She says that as time goes by, I become more aware of the constant changes affecting our planet Earth. Experiencing these changes has revealed to me the need to understand and find ways to save marine ecosystems from extinction. A tall order, but go for it. You have the energy of the youth. So go ahead, Maria, you take it away.
So good afternoon, everyone. My name is Maria Plapral, and I'm an intern for the Summer Undergraduate Research Fellowship through PICASC. and today I'll be presenting the effects of marine heat waves on coral skeletal density in Hilo in Hawaii. This experiment has been done with the help of Dr. Steve Du, Megan Carney, and Matt Connolly from the University of Hawaii at Hilo. So first of all, to get to know me better, I'll tell you a little bit more about myself. I'm originally from Barcelona in Spain. I'm a student at the University of Hawaii at Hilo, majoring in marine science and pursuing a certificate in data science. In the future, I'm interested in becoming a coral reef conservationist and hopefully work at my dream place in French Polynesia. So I'll start by explaining what marine heat waves are, and more specifically, what was happening in Hawaii. So marine heat waves are periods of anomalously high ocean temperatures over a time period. So we have this first graph here, which show us on the x-axis the date with the time period from 2015 to 2017. And then on the y-axis, we have the sea water temperature in degrees Celsius. This research site was based in Hawaiʻi Island in Hilo, in the east side of the island. And the red line shows the historical mean for that specific site. As we can see, the majority of our data points are within that historical mean site. So that shows like a typical map of what the temperatures will be from a whole year over 2 years time period. Then we have another, the same graph, but this time the research site is based in Kauai, which is situated also in Hawaiʻi Island, but on the west side of the island. We can observe that in September 2015, there was a major peak where the temperature reached 31 degrees Celsius, and that was the major bleaching event in Hawaii, where 80% of the corals bleached. So then we have this heat map with the analyzed sea surface temperatures in degrees Celsius from the island of Hawaii. So on the bottom, we can see a color scale with red colors showing temperature range from 29 to 30 degrees Celsius. And then we have more like green and blue colors from temperatures ranging from 27.5 to 28. And then blue and purple colors with temperatures from 26 to 27 degrees Celsius. So here we can clearly see like a big warm mass of water on the west side of the island right here, where Kauai is situated. And then we can see that on the other side of the island, on Hilo side, it is more lower temperatures with like more blue and green colors with temperatures ranging from 27.5 to 28 degrees Celsius. So coral bleaching. So as I mentioned before, marine heat waves are becoming they're happening more often. And we studied coral reefs as our organism. Coral reefs are among the most vulnerable ecosystems to climate change. And what happens is like once marine heat waves start happening, coral reefs start experiencing what we call thermal stress. And then they start, one of the first visible signs that we can observe are bleaching. Bleaching is the symbiosis breakdown between corals and their dinoflagellate algae, Symbiodiniaceae. So here we have a little illustration. So we can see a healthy coral. And then once they start experiencing thermal stress, they start releasing their dinoflagellate and they start leaving the tissues of the living coral. And that's why they lose their colors and they bleach. So marine heat waves are more frequent and severe, and coral bleaching events are predicted to increase as time goes by. So one of our purposes of our experiments was then to find a tipping point in which a tipping point in the physiological performance of the coral holobiont to a mosaic of thermal stress. I also want to mention some previous studies. So this is the Hawaiian Islands. Some of the studies have been done in Oʻahu, for example, where they monitored bleaching responses of Mantipora capitata and Paraitis compressa during marine heat waves. Another study was in Maui, where they tracked the fate of bleached corals after the 2015 bleaching event. And then there are some studies that have been done in Hawaii, but they have been done on the west side of the island, where they studied the bleaching and catastrophic mortality of reef building corals. So another purpose of our experiment was also to expand the location where the marine heat waves experiments have been done. So we based our experiment on Hilo side, on the east side of the island. So our study site, as I mentioned before, we were based in Hawaiʻi Island in Hilo, and we work at the Pacific Aquaculture and Coastal Resources Center, also called PACRAC, which is located right here. This is a zoom-in image of the aquaria facility, and where the arrow is pointing is where we worked. We also collected coral fragments from Onekahaka Beach Park, located right there. So experimental design. So this is a plot that we've made to explain what our experiment was about. So on the x-axis, we can see the duration in weeks, and on the y-axis, we can see the intensity in degrees Celsius. Each point represents a tank, and then what we try to do is simulate a marine heat wave for each tank. So based on the duration and the intensity. So for example, we had a control tank, which was this cross right here, which was set at 24 degrees Celsius for the 4 weeks, for the whole experiment. However, every tank was set at a different intensity and for a different amount of time. For example, this one was set at 25 degrees Celsius for 1 week. And then after the first week, the 3 other weeks, it was dropped to 24 degrees Celsius, which was the control tank. So as you can see, each cross corresponds to a tank. And we have also a color coding for that corresponds to the degree heating weeks that I will explain later on. So our first step was building a coral nursery. We started by plumbing a system. So our system is based of 16 tanks. So we had to plumb all the water that was sourced from underground. And we had 16 tanks with a flow-through system with water coming inside and then out through this sump right here. The photo is very, very, very messy, but it was very chaotic, but it was amazing. Each tank had approximately 6 corals in it, and it was equipped with a water heater and a water pump. Then the next step was coral collection and fragmentation. So we went to Onekahaka Beach Park and we collected around 22 small branches of corals of opportunity from the species Montipora capitata. What I mean by corals of opportunity is that the corals were already broken either by human interactions or by wave energy. So we just collected them and tried to use them for our experiment. We then fragmented them into 100 pieces. They were pretty small, only 2 by 2 centimeters, and we glued them to tiles using aquastic epoxy putty. So the first image, we can see we're using a saw to cut the pieces. And then on the bottom, we can see we're trying to glue them to the tiles using the epoxy putty. Expected results. So, sorry.
Go ahead.
Okay, so we start by trying to imagine this plot. So on the bottom, on the x-axis, we have the degree heating weeks. Degree heating weeks is the amount of thermal stress accumulated on an organism over a period of time. So I calculated that by calculating the difference in temperature for each tank for each day. And I calculated the difference from the control tank, which was set at 24 degrees Celsius. And then I added all the differences in temperature, and I also divided it by the number of days in a week. And our experiment was only done for 2 weeks, not 4 weeks. I'll tell later why. And so then I calculated the different degree heating weeks for each week. On the y-axis, we have the days it took for 50% of the corals to bleach. So then our null hypothesis was that we assumed that there was no change in time to bleaching with increasing degree heating weeks. However, if I ask this question, does increasing thermal stress cause bleaching rates to increase? You would think it does, right? So we obtained this negative correlation between the degree heating weeks and the days it took for 50% of the corals in each tank to bleach. So because as you increase the degree heating weeks, you would expect that it takes less time for the corals to bleach, right? But if you make it shorter degree heating weeks, like lower, you would take more time to bleach. However, our results don't really tell us the same thing. So this is also the same kind of plot. On the bottom x-axis, we have the degree heating weeks. And on the y-axis, we have the days it took for 50% of the corals in each tank to bleach. As you can see, there's not really a correlation between both of them. And on the top, we also have a scale, color scale with the intensity for each tank. So yeah, as you can see, there's not really a correlation between both of them. So we don't think that degree heating weeks is a good indicator to predict the bleaching rate in this mosaic of thermal stress. That might have been due because we realized that our levels of dissolved oxygen were very low. Our levels of alkalinity were also very low. That's why we had to stop our experiment after 2 weeks, and then we have to restart it. So now it's more continuous work that we're going to be doing. Future directions. There's also some non-intrusive techniques that we also used. So we use 3D photogrammetry. So for example, this is a photo of a 3D model that I created of a coral fragment. So we took around 50 images per coral fragment, taken at different angles, using an Olympus underwater camera. And then we used the software Agisoft Metashape to create the 3D models. And then we also used another software called Cloud Compare, which will allow us to scale the coral fragment and measure the volume. All this work has been done with a collaboration of the Megalab. And we also use another non-intrusive technique, which is called the buoyant weight technique. This technique involves weighting the corals while they're suspended in a medium buoyant water. So this allowed us to weight the corals and find their mass. So once we have their mass and their volume, we can find the density of the coral skeletal. And then we can use that to measure coral growth rates and to assess any impacts or recovery impacts on the rates of growth of the corals. I also
got a real hook on it.
I also
I
wanted
might probably pull the stitch out of the needle.
Sorry.
I also wanted to mention that you can my title was like the effect of thermal stress on coral skeletal density, and I really didn't talk about that much because this is like ongoing projects, so we're still we got some data on buoyant weight technique, but we're still going to continue working more on the 3D photogrammetry, and everything is linked, but I emphasize more on marine heat waves because that's the main cause. So a little reflection. I really want to thank PyCask and Hawaiʻi Island for this amazing opportunity, and of course my mentor and all the people that helped me. It was amazing because I learned scientific methods. I also learned that with dedication and passion, you can really achieve everything you want. It was a lot of hard work and hands-on field experience, like going collecting corals, fragmenting them, working on a lab. It was very cool. I also had the opportunity to network with other people through the workshops that you guys organized. And I also learned that communication skills are very, very important. So thank you so much to Dr. Steve Du, which was my mentor, Matt Connolly and Megan Carney, for all their support through the whole experiment. And I also want to thank PyCask, Hawaiʻi Island, and the University of Hawaii at Hilo. Thank you so much.
Do we have any questions?
I'm super interested in growth rates. So I was super stoked to see that you put that on the slide. Do you have any expectations for what you would see in growth rates if you're trying to use them as an indicator of a bleaching event before the bleaching event happens? What do you think might happen?
I think it would decrease the coral growth because they're under so much thermal stress that they're just trying to like survive as they can. They're not like even trying to grow. But we did get some data from before the experiment, after the experiment of their mass, and they did grow. So they can grow under some thermal stress. So that's really cool, I think.
Anything else? Yeah, one more.
Great talk, Maria. Really enjoyed the slides. You said at the beginning that you were interested in coral conservation. So curious what your experience or what you thought coral conservation consisted of before, and how this experience influences that decision, and what you feel like you'll need to bring with you to pursue that career.
That's a very good question, because I realized that being a researcher is not very easy. It was a lot of frustration and like, yeah, for becoming a coral reef conservationist, I think you have to keep working on that passion and dedication so you can really achieve that. And then also think about the future. So why you're doing all this job and what's your goal in all this. So I think I'm going to continue working on that.
Okay. All right. Thank you very much. So we've reached the halfway point. Would people like a brief bathroom break, or shall we plow ahead? Yes, bathroom break. Okay. Come back in 4 minutes and we will get back on track with the next talk. And those online, don't go away, just come back in 4 minutes. Yes, so we are moving on. We are shifting to different invertebrates. Maddie Morales is a rising junior at the University of Washington, studying marine biology, and another Hawaii Sea Grant Surf Fellow. Maddie says, I've always been extremely passionate about marine invertebrates and what many consider to be underappreciated organisms that play little known but incredibly important roles in our world's oceans. So why don't you tell us a little bit more about forams?
I will.
All right. There's the clicker.
Okay, so this is my talk, I Kawa Mamua Kawa Mahope, Conservation, Paleobiology, and Palmyra Atoll. And what my title basically means is the past is the key to the present. And it pretty much means that we can't learn about our future and our present without looking to the past for answers. And as I go through my talk, I hope that this title begins to tie in with the project that I'm working on. So it doesn't take a degree in conservation biology or wildlife biology or biology in general to know that the current state of our world's biosphere is in complete disarray. Every day we're losing more and more biodiversity across all groups of taxa, whether it be marine, terrestrial, or freshwater. And so modern conservationists are kind of scrambling to both sort through data and collect data that could potentially give us a good idea of what to shoot for in terms of saving or restoring environments and populations. But because of the novelty of conservation as an important and vital field of research, the record of the data that we have pertaining to organisms and environments and things like that is very shallow. So this brings us to how typically people talk about conservation, which is in the present or future context, where we need to focus on preventing the worsening of decline now and in the future, but also about going back and preserving and restoring communities to previous states. But this also begs the question of like, what does a previous state look like? And this is a very difficult question to answer for a number of reasons. The main one being that anthropogenic climate change affects ecosystems and communities on a decadal to like 100 decades to hundreds of years timescale. And most of the data that we have, like that's widely available, is on the scale of 1,000 to 1,000,000 years from traditional paleontological data, or it's on the scale of days to years from modern biological and ecological data. And so what we're missing is that data, like I said, like on the decadal to century scale, and that's where we have invisible time scales, or like that kind of that gap between the really old stuff and the really new stuff. And that's what we really need in order to like work on conservation. And so this disparity in appropriately scaled data means that conservation as a field has frequently run into the problem of like, where do we conserve to? Is it like 100 years ago? Is it 200 years ago? Is it 20 years ago? And if we eventually do decide on like a baseline, then like, what do these communities even look like? Because like I said before, the data that we have going back into like hundreds or 200s of years or like tens of years is very bad. Most of it only goes back to around 1970. And like, even then it's like really spotty. So this is where conservation paleobiology comes in. And that is where what we can use to construct pre-disturbance, pre-anthropogenic disturbance, ecological, population, environmental pictures, since, like I said, the scope of the data that we have is so incredibly shallow. However, in places like the Pacific Ocean, we are missing a lot of data. And this is a little map showing all the paleoecology data points from the past 2.58 million years. And as you can see, most of the data points are concentrated in North America and Europe, with most of it being in the United States. And there's not a whole lot going on in the middle Pacific. There's probably around like 5 or 6 data points in total. And this is a really big problem because the Pacific Ocean is quite literally the largest ocean basin on the planet. And it's also home to a wide variety of communities and cultures and environments that are beginning to face the very first impacts of climate change. And so in recent years, we have been seeing a push by different agencies such as the USGS and the U.S. Fish and Wildlife Services to do more historical data collection in the Pacific. And this is where the focus of my project comes in, which is the atoll Palmyra. So Palmyra is a small mid-Pacific atoll just south of the Hawaiian Islands, and it's a really good place to do this sort of data collection in the Pacific. And it's a site where the USGS and the U.S. Fish and Wildlife Services have been collaborating on for like almost 2 decades. Because of its location, and also because of its shared history that it has with modern human activity. So Palmyra is a mid-Pacific atoll, like I said, and like Hawaii, it was deemed strategically favorable by the United States military during World War II. And because of that, it was heavily modified in its physical environment to suit the needs of the military. So this is a picture of what Palmyra used to look like in 1942. And on the right, there's a map that gives kind of like a general overview of the shape of the island, like the toe and like the silhouettes of the islets and things like that. And the present day one is on top. That's what it looks like now. And what it used to look like in the 1930s is down below. And as you can see, the military construction resulted in a lot of dredging, a lot of creation of land bridges and islands, and a lot of conjoining of islands together, which has overall resulted in a restriction of water flow into the system, as well as some other untold and effects that we're looking at right now that affect like the biology and the physicality of the system as a whole. So in order to do that, the USGS has been, that's what it looks like now. In order to do that, the USGS has been doing a lot of coring, like sediment coring surveys. And this is a picture of the most recent one that was done in 2024. So just this year, that's the core. It's in a metal tube. But the core that we are working on for my project is from 2009. So this is a photo. Well, this is only a part of it. So the stuff at the top is the very newest part of the core. And then like as you go down, it gets older and older. This is only a section of it. The entire thing extends to about 3.2 meters. But this portion, as you will soon come to find, is the most relevant and important. So on the right, there's an X-ray photo, and then on the left, there's a color photo. Both are of the same portion of the core. And the effects of World War II land construction are very clearly preserved, as you can see in the X-ray. So the middle section, that lighter part, is the World War II construction horizon. And then before that is the pre-World War II sediment, and it's heavily mixed, which was likely due to the increased amount of water flow that came into the system, as well as increased bioturbator activity, which is like worms and things that burrow into the sand. And then after that, we have the post-World War II construction horizon. Those sediments are very heavily layered, which is a direct result of the lack of water flow that comes into the system because of the construction. And so all of this as a whole makes Palmyra and this like core specifically very good for deriving data about how human land use changes affect marine ecosystems in the Pacific. But in order to get that sort of data, we decided to look at forams or foraminifera community diversity. And so forams are basically like these little, they're really tiny. They're like specks of dust. They're pretty much amoebas with shells, if you know what amoebas are. They're very good at collecting, they're very good for collecting paleobiological data because they're extremely small and they're also very sensitive to changes in water quality, oxygen content, and other biotic and abiotic environment changes. And not only that, but all of those changes are preserved in like the shells that they produce. And on top of that, they're fairly common throughout the time scale in sediment cores that are taken from Palmyra and as well as like everywhere else in the ocean. So in order to get this data, we will go through our methods. And basically what we did is we set out a microscope and we pick through this tray of sand, and using the tiniest possible paintbrush, we pick the forams out and isolate them. And then we put them on a slide to arrange them based off of their morphological characteristics because there's no genetic material, and the only thing we can do is like group them based off of how they look. And then after that, we used our $1,000 multiple tally counter to count the species. And then we put that into a data sheet, and then we have our species guide. But before we know what species to like count them as, we have to use our identification guide, which, as it turns out, was completely non-existent before I started working on this project. So that's like the main product that came out of this was this very spotty identification guide for forams of Palmyra. And it's extremely important because since we're looking at species diversity, we can't quantify diversity without knowing what species are present. So that's why the ID guide is so important. And also knowing what species are present is important for knowing like the environmental qualities that were present in the past, now, and potentially in the future, because forams are really sensitive, like I said before. So only certain species can exist under certain conditions, like some are tolerant of lower oxygen or lower nutrients, etc. But again, this was very hard because foraminifera taxonomy is extremely complicated in that there's no standing authority on what designates a species. And again, there's no guide for Palmyra, so I kind of had to use my intuition and whatever was available online. And that proved to be very difficult because most of what we were seeing were very cryptic. They were unnamed, and apparently they were very region specific because I couldn't find them anywhere else. So yeah, and that is what leads us to our data. So without that, we couldn't have constructed our pilot data, and which is like visualized in front of you here in a stacked bar plot. So this plot shows foram community structure pre and post World War II. So the pre-bin is on the left, which is the one with the more colorful pattern. And then the one on the right is post World War II. And in the pre-World War II bin, we can see that we have way more abundant dominant taxa, but not a very high species richness, since most of the present forams can be classified in one of those 8 colorful bars, with a remainder being left in the rare species group. And rare species basically means that there's like maybe one of that species left. Oh, sorry.
They were scared of a marked house. They thought that would help define carnivore to the middle. You see a lot.
Anyway, so so in contrast, the post-World War II bin consists mostly of these rare species that we've kind of like lumped into one category because like there's only one of each. And that is interesting because traditionally in ecology throughout the ages, we have tended to coincide species richness with ecosystem resiliency. But now we know that this is not exactly the case, and that redundancy, which is when you have lots of species with lots of overlapping niches and functions, that is what coincides with a healthy, resilient ecosystem rather than species richness. And some more food for thought is that the rarity of a once abundant species is thought to be a harbinger for ecosystem collapse. And that's interesting because, as you could see previously, a lot of the abundant taxa that was once present pre-World War II are no longer present, and instead we just have like this really high species richness. Like there's a lot of like one-off like little tiny species that are present instead of having like a healthy community of abundant dominant taxa. But because the data that we have is very pilot, it's preliminary, it's very initial, we can't exactly say what's happening, but it's pretty safe to say that all current signs point to there having been a very major ecosystem change in the advent of the World War II construction horizon, which brings us to our future work. So using the taxonomic guide that I'm working on, we are going to further study the way that foraminifera community changes have occurred in Palmyra. And this is just all to say that the data that we're collecting isn't just for the sake of like picking through sand and looking at cool organisms, which are very cool. But it's all part of a very large project and movement within the USGS and the US Fish and Wildlife Services to develop better climate and biotic records in the Pacific that will hopefully better change and drive science and conservation efforts in those low-lying Pacific communities going forward in the face of climate change. And again, so going forward, we will continue to look at those changes, and it will eventually give us a better, more solid, quantifiable idea of the changes that have happened in Palmyra in the advent of that World War II construction horizon. Thank you.
Very cool. So we have time for one, maybe two short questions.
Yes.
Travis?
It sounds like a project for artificial intelligence, because you don't have standard guides. We have a lot of, this sounds like a project for AI to help with the taxonomy, if you don't have standard guides for identification.
Yes, it is actually. My mentor, Sada, their PhD project actually had a lot to do with foraminifera that are present in California, and AI was used a lot, and it was a very big help in classifying and like working on the taxonomy of these organisms, since most of what we have is like there's no genetic material, like there's no soft tissue, it's purely morphological. So yes, going forward, I think it would be very helpful to somehow develop like a model, like I don't know anything about computers, a model to classify things. But yeah, it's good food for thought. Increasing efficiency. Yeah, we're working on it. Yeah, maybe. I don't know.
Lillian, you had a question. Do you want to unmute yourself and ask it?
Yeah, can you hear me now?
Yes.
Okay. Very interesting. I was curious if, in addition to patterns in diversity, if it's possible with forams to look at patterns in like max size. Is there any evidence, for instance, that like the same species foram used to be bigger but is now smaller, or like some way to track growth rate in addition to size? And in that way, you could maybe try to understand if like the actual health of the ecosystem has changed as opposed to just the diversity.
Yes, that is a very possible application of like foram growth sizes and things like that. Forams are very sensitive, and they're also very variable in terms of like how they produce their tests or like the shells that they make. So under certain conditions, certain species can grow larger or smaller. They can develop like additional chambers is what they're called. Like they can look like completely different species just depending on like the oxygen load or the turbidity or even like the nutrient availability. So, but again, it all comes down to being able to identify them by species, or at the very least by genus, which is also very difficult. So if we don't have that information, then we can't necessarily say like how they're changing. Yeah. Yes, and we are going to do that. Yes.
Sounds like you have an entire graduate career paved for you.
Yeah.
If you come back here.
Okay.
Great. Well, that's fascinating. Thanks very much.
Thank you.
Can I take your mic? Mike.
Oh. Thanks.
Okay, moving on. We are now going to shift to something completely different, some engineering. So Erica Kui, changed. There she is our last Hawaii Sea Grant Surf Fellow, and she's a rising sophomore at MIT studying mechanical engineering. She shared that she is, quote, fascinated by the intersection of engineering and ocean science because it allows me to apply my passion for mechanical engineering to sustain, protect, and understand the ocean. So why don't you share with us a little bit about that, Erica?
Hi, everyone. My name is Erica Kui, and I will be presenting on wave energy conversion and its applications in Hawaii. My mentor was Dr. Patrick Cross, and I also worked with Kimball Milliken and Dr. Troy Heitman, as well as their team at UH. So I think that wave energy has a lot of applications in Hawaii and coastal communities because the ocean holds so much energy in it. And if you compare it to other types of renewable energy sources, such as wind energy, water has a much higher density than air. So you can use smaller devices in order to get more energy from it. Also, the waves are very predictable, and waves are always moving and pretty consistent. So it would be a pretty reliable source of energy compared to solar and wind energy, which depend on the weather. There are also some challenges of wave energy, such as the cost of doing research and testing, materials, deployment, and maintenance. And also when you're designing to put something to put in the seawater, salt water is very corrosive, and it takes a lot to design something that will be able to withstand salt water, and also just the environment in the ocean can be very forceful. And there are also challenges with infrastructure in connecting to the grid once wave energy converters are deployed in the water, and also having infrastructure offshore to support deployment and maintenance in order to make that process easier. Wave energy converters can also have an environmental impact, such as on marine life, such as fish, and they can also make noise, which could be disruptive to underwater ecosystems. So here are some examples of wave energy converters. So the first one is an oscillating water column, which is made up of a device that sits on the surface of the water. And inside is an air chamber. So when the waves move up and down, it pushes the air, which then spins a turbine, and then that can be connected to a generator to create electricity. Another example is a flap type energy converter, which is made of a flap or a panel, and it's connected to the base at a hinge. So then it will pivot along with the motion of the wave, and then that is then turned into energy. And then another example is a turbine. So similar to a wind turbine, it's basically just in the water, and the waves or tidal energy will push the turbine to generate electricity. And on the bottom is an example that Kimball put together of a cycloidal rotor type of turbine, which can spin, and it's cool to see how the waves will actually make the blades spin pretty fast. So putting wave energy converters in the ocean could affect the environment because of the noise that it can produce. So one way of monitoring noise levels in the ocean is by using a sea spider, which is equipped with hydrophones in order to collect data on the acoustic levels in the ocean. So I helped to assemble the hydrophones and also set up the acoustic release mechanism, which is used to be able to retrieve the sea spider from the ocean after months of collecting data. And then that data can then be used to see how the devices in the ocean can impact marine life. I also participated in a survey at WETS, the Wave Energy Test Site in Kaneohe. We took a boat out, and the purpose of this survey was to inspect buoys and their underwater systems. So we also took an ROV, or an underwater robot, to collect images and videos of the buoys in order to make sure that nothing was corroded or broken. And then this is the Hawaii Wave Surge Energy Converter, or HAWSEC. It is a flap type wave energy converter. As you can see, there's a rectangle, and at the bottom is a hinge, so then it'll flap back and forth when the waves push and pull it. And the next step for this project is to eventually scale it up, so it'll eventually be deployed in Mackay Pier. So this is the bench test setup for HAWSEC. Before putting it in the water, it's important to test the components outside of the water in order to understand the whole system and see how much energy it could produce. So in this photo, you can see that there's in the back is a linear actuator, which moves back and forth linearly. And that is connected to a hydraulic system, which pulls water from the tank, and the water comes out and pushes or spins the turbine at the top, which can be connected to a generator to produce electricity. So the next step for this project is to scale up the bench test setup. Eventually, they want to use a larger linear actuator, which would provide a larger amount of force of 23 kilonewtons. So my task was to design a bench test setup that would be able to withhold that amount of force. So I started off by looking into possible solutions, such as just like a simple table design or structure that would be able to support a large amount of force. And I eventually found the hydraulic press design, which is a frame made up of C channels, and it's designed to withstand a large amount of force. So this is the CAD model that I made. It's made up of C channels and is similar to the hydraulic press design, but just mounted horizontally. And you can also see the linear actuator and cylinder. So I used this model to create a simulation, which would allow me to understand how applying a 23 kilonewton force on the structure would affect it. So from the simulation, you can see the deformation of the structure and the displacement and stress throughout it. So this allowed me to make comparisons between different versions of the structure that I made using different types of beams. So I compared using C channels and rectangular tubing of different thicknesses. And from the simulation, I was able to determine which one would be the most optimal to use in this bench test setup. And then lastly, I compared materials that were pretty common to use for these beams. This is just a way for me to understand the differences between materials such as steel and aluminum, and in order to decide which would be the best to use for the bench tests. So this summer, I was able to see the deployment of 2 wave energy converters in Hawaii. And so we went out to the wave energy test site. And this is the first WEC, which is a C-ray by the company Sea Power. It's pretty small and is used for low power needs, such as powering underwater vehicles. And it's made up of 2 floating buoys, which will move around in the water, and that pivoting motion is then translated into energy. And then this is the second wave energy converter called OE35 by Ocean Energy. And it's a much larger wave energy converter and is used for more commercial scale. And it can make up to 1.25 megawatts of power. So the OE35 is an oscillating water column type of wave energy converter. So on the inside, there's a chamber, and when the waves move up and down, it'll push air, and then that will spin the air turbine, which generates electricity. Yeah, I think it was really cool to be able to see these two wave energy converters deployed this summer, especially seeing how much work and how many years of testing and designing goes into making these wave energy converters before they can finally be deployed and eventually used to supply power. So in the future, for these projects, we would need to continue with testing and scaling up models in order to understand how much energy can be produced, and also scale it up to be able to produce more energy. And also in coastal communities and in Hawaii, we would need to expand infrastructure in order to support the wave energy converters and be able to supply power to homes. And I would like to thank Dr. Patrick Cross and his team for introducing me to the field of wave energy, because it was really cool to see the projects that they're working on. And also, I think in the future, I would be curious to learn more about wave energy projects going on around the world, because there's so many different approaches to this technology. Thank you.
All right, we've got some time for questions. Anybody have any?
Erica, on your bench test, did you have a conclusion on what shape the materials were the best?
Yeah, I eventually decided on using a rectangular tubing of like a 1/4 inch thickness. And then for material, generally, like all of the steels would work pretty well.
Maddie, you also had a question?
Yeah. When you're designing structures that generate wave energy and you're putting them in the ocean and everything, like how do you take into account organism settlement like on the structures that you build? Like, is that something that you think of right now, like in the process, like at this point in the process, or is that kind of like an afterthought?
I think it's like definitely important to consider like how it'll affect like marine life and organisms, and that's something that goes into like the testing of the wave energy converters. Like I think generally like when you're designing it, there's a lot of testing that you can do in order to make sure that it doesn't like affect fish.
Yeah, when designing it, do you also look at like material composition with regards to like corrosion and how the ocean's going to affect it and like different salinities? Do you think about that and how that could affect the future composition of it and then maintenance of it going forward?
Yeah, it's definitely important to use materials that will be able to, that won't like corrode easily in salt water. And then also like with maintenance, just like checking up on the devices to make sure that nothing's broken or, and yeah.
I also wondered, going back to that idea of the critters settling on the machinery, could that actually have an effect on the efficiency of the systems? Because if you've tested it with nothing growing on it, and then you have, say, I don't know, corals or barnacles or something that start growing on the machine, that could change how much it swashes back and forth or whatever mechanism you actually end up doing.
Yeah, I think that could definitely impact like the effectiveness of it. But I think that's something that like you would need to do with like maintenance in order to like check up on it often to make sure that nothing's like settling on it.
Okay. All right. Cool, different look. Thank you. Okay, now we are going to shift from the water to the earth. We now have our final two talks, which are both terrestrial ecosystem focused. The first one is by Jaslyn Mura, who is a PICASC Surf Fellow She's an environmental science and resource management major at the University of Washington, entering her senior year. Jaslyn says that she is passionate about climate change, as she's seen how its effects have seriously impacted our home. She hopes to share the results of her project to spread awareness within her Hawaii Island community about climate change and its impacts. Here's your mic. You may click it on.
I think I'll be okay. Okay. Hi, everyone. My name is Jaslyn Mira, and this summer I had the privilege of working with research ecologist Dr. Helen Safair, and we used R to help us perform an analysis that helps us understand Hawaii's biomes in a changing climate. So to begin, I kind of just want to explain why we conducted this analysis, and it's all related to the fact that climate change is causing powerful ecological transformation, and our historical and normal climate conditions are not being maintained around the world. And this is causing our biomes to shift, and we don't know what biomes can be maintained. And as we ask these questions of what's happening to our biomes, we can implement this new decision-making tool called the Resist, Accept, Direct framework. And basically the idea of this framework is to move decision makers away from the traditional approach of focusing on one singular outcome for a place. And to do this, RAD recognizes that our future holds many different climate scenarios, which also means that there are a range of possible ecological conditions for any given place. And given these possibilities, managers can then decide to resist, accept, or direct change. However, to even implement this framework, we have to evaluate what ecological conditions or outcomes are possible for a place. And this is exactly what our analysis does. So our analysis utilizes temperature and mean annual precipitation for our climate conditions, and these factors play an important role in the type of vegetation that can be found in a biome. The analysis also takes climate conditions from a historical and recent time period, and it also includes a projection of future climate conditions. And by using these time periods, we can see how the biomes have shifted in Hawaii over time. And within our analysis, we also investigate the extent of grass cover, as it's a major contributor to wildfires, which therefore accelerates changes in a biome. So overall, these factors play a role in what biomes can be supported in a certain place. And again, we're interested in determining the range of biomes that are possible. So first, we'll take a look at this chart that was made by Robert Whitaker, and it displays how biomes are related to each other given precipitation and temperature. And so here you can see that the biome boundaries are very sharp and discrete, and this graph doesn't really suggest that climate conditions can support multiple biomes, meaning if you look at the gold stars, you see that you'd expect to find one or maybe two different climates at these points. So, however, in reality, the biome boundaries are not as neat. And here we see, after replicating Whitaker's diagram using biomes found here in Hawaii, we have a better visual understanding of how the biomes are actually related to each other. We can see that there are cases highlighted by the red circles where the biomes actually overlap each other. And overall, this diagram is showing us how, given different climate scenarios, multiple biomes can be supported. Okay, so to go into a little bit more detail with this diagram, basically what we're doing is we're plotting all the biomes in the state of Hawaii, and this is done as each point represents a location in Hawaii. So next, we can define a climate scenario by determining a small range of temperature and mean annual precipitation, which is defined by our little black box there. And then we can zoom in on that box and determine the different biomes that can be supported in that given climate scenario. And our stack bar chart on our left shows that shows us the proportion of each biome that can be supported in our defined climate scenario. So for our analysis, we defined different climate scenarios by creating climate bins, and this was done by dividing our temperature scale by 0.5, and we applied a log to our precipitation scale as changes in precipitation has a greater effect on climate than changes in temperature. So in this figure, our bins are represented by the grid, and by looking at these bins, we can match all the locations in Hawaii represented by the points to a climate bin and then determine the proportion of each biome within that climate scenario. So here again, what we're doing is we're matching the points that represent a location in Hawaii to a climate bin. But instead of looking at each individual climate scenario, we can look at each biome, which is shown, we can look at each biome to understand what conditions match a type of biome. And this is shown in the darkest shading of green, as it indicates a higher proportion of climate matching the given biome in each panel. So another way to think about it is if we think about our Whittaker plot that I showed you in the beginning, if he were to create a biome plot again from these panels, he would only take the dominating conditions, so the darkest shade of green, and create his plot. But instead, what we're doing is we're mapping out all the climate scenarios that are compatible with the different biomes, which is shown in the lighter shading of green. And we do this to display a wider range of possible climate scenarios that can support each biome here. So as we went through this project, we wanted to focus on some areas in Pu'uwawā. These points are Makai, Northeast Henahena, and our forest bird points. And basically these points represent a steep climate and precipitation gradient, as our Makai point is our driest and hottest area, and our forest bird point is our wettest and coolest area. And here we also include a map of the predicted historical biome cover. And this is before human influence. And we're focusing on these points so that we can look at how the biomes in these areas have shifted over time. And also, hopefully, this gives people better insight to what our analysis is doing, as the points may be familiar to some people who are from like the Big Island. Okay, so now looking at our focal areas, we've identified the biomes that are the best match for the climate conditions for our focal areas over time. So our focal areas are denoted by the different shapes of the points, and our climate conditions during different time periods are denoted by the colors. So here we see that with historical climate conditions, our forest bird point and Northeast Henahena point best match our mesic forest biome, as the points are found in the darkest shade of green in the mesic forest panel. Then as we look at our recent climate conditions, we can see that our Northeast Henahena point and forest bird point best match our dry forest biome. And again, looking at our future climate conditions, those points are still a best fit for our dry forest biome. However, if you take a look at the red star in our dry forest biome, we see that it's moving out of our climate space. And basically what that means is it's moving into a climate bin that we haven't created. And unfortunately, that means that we don't know what biomes will be supported by those climate conditions. Okay, so to analyze our focal areas, what we did is we took our climate scenarios or bins that are found in each of the focal areas and then determined the proportion of biomes that can be supported within the climate of these areas. So here we see our forest bird chart tells us that historical climate conditions supported a mesic forest biome, but over time, with climate change, we expect the conditions to no longer support a mesic forest biome, but instead dry forests and dry shrubland. And this is similar to our Northeast Henahena point, where historically conditions supported a mesic forest biome, but now conditions have moved the system to conditions that are only supporting a dry forest biome. So after we created different climate bins representing varying climate conditions, and scenarios, and also analyze the proportion of biomes within each bin, we can map the climate analogs of each climate condition for our focal areas. And basically what we're doing is identifying places with similar climate conditions to our focal areas. And by doing this, it will give managers a existing place to further understand the impacts of the specific climate conditions that we're concerned about. So here we can compare the predicted historical biome cover to a map of land cover now. And here this alluvian chart shows us how the biomes have shifted. And by following each path, we can see how the biomes have changed due to human modification. Here we're highlighting the proportion of biomes that have shifted into grassland, which is represented by our orange path. And we did this because we're concerned about invasive grass, because as I've said before, it's a contributor to wildfires and can accelerate dramatic biome shifts. So focusing on invasive grass a little bit more, we can look at this figure, which again shows us the proportion of climate that matches the type of land cover. And here these panels are highlighting forest cover and grassland cover. So the darker shading indicates a higher proportion of climate matching the given type of land cover. So looking at our forest panel, we see that the shading of the bins is much lighter, which indicates that climate is less of a match to forest cover. And if we take a look at our grassland panel, specifically focusing on our focal areas, we see that our focal areas over time are moving into a climate bin or scenario where the proportion of climate is almost to the point of completely matching grassland cover. And this is concerning as it suggests that these focal areas may be at high risk to invasive grassland cover. Okay, so now that I've walked through this analysis, I hope it shows you how we can step away from the idea of being set on a singular solution or outcome and instead recognize the varying possibilities for place. And by using this analysis and visualizations, managers can decide what type of management within the RAD framework is feasible based on how the code identifies the proportion of biomes given a climate scenario and the mapped analogs that we've done. And these management options being resisting change, accepting change, or directing change. And basically in a world with changing climate, I think it's really important that we recognize that transformation is happening. However, given that, I think it's also really important to know that there are a range of outcomes that are possible, and therefore different management approaches that we can take. And so while this version of our analysis really only focused on Hawaii and Puʻuwawā, we can use the same methodology on a global scale, which is exactly what I'm doing in our last week of my fellowship. And this will just help us understand the ecological variation around the world and also help us map different climate analogs around the world with climate conditions that we're concerned about. And to finish us off, I'd just like to thank my mentor, Helen, for all her guidance and support that she's provided me throughout this project. And I'm extremely grateful to PICAST and Hawaii Sea Grant for giving me the opportunity to work with Helen. And thank you all for listening. And does anybody have any questions?
I think we have time for a question. Anybody got it? Or anybody online have a question? Lillian again. Go ahead, Lillian.
Sorry, if anybody else wants to ask a question, that's fine.
Nope, you're good.
Great. Yeah, that was really, really interesting. I live on Big Island, and so I've been to Puʻuwawā, and I know that that's a location where there's a fair amount of like plant restoration and outplanting happening. And so I was curious if yours and Dr. Sofar's projections are kind of like causing the restoration efforts to change? Like if you go to a previously wet site, do you know if people are outplanting plants that prefer more wet environments, or are people starting to transition into maybe planting species that will do better in like these predicted mesic habitats that we assume that location will change to?
Yeah, I don't think we've like taken this code to managers yet to like have them implement it into their work. But I think that's the really cool thing about the code, where it shows you what biomes are possible. So like you said, managers can change the type of vegetation that they're trying to conserve in an area, which is what's related to the resist, accept, direct framework, where you can decide to resist change, where you're trying to keep the area as a mesic forest, or if you're directing change where it looks like the system's going to move to a dry forest, and that's the best option instead of like it becoming completely invaded by invasive grass. Then I think the code really helps you see like what options or outcomes are possible for an area, if that answers your question.
Yes, thank you.
Okay, oh, Travis, you have one more?
So biomes is a tough concept to apply to Hawaii because huge changes in climate occur with very small geographic ranges, unlike biomes that are meant to be these very large scale systems. I mean, to me, it seems like there's an advantage of having a mix of plants and communities in a space so that they can kind of adapt to that. I don't know if that's come up at all. It's like not just sort of saying this is where you're going to be in 50 years, but rather we need to have a large suite of species in that area because they can be quite variable over short distances as well.
Yeah, I think that's kind of what I've been interpreting the analysis as to, where it's like the stacked bar charts, which shows you like the proportion of different biomes that could be found in an area. I think you could take that and be like, oh, well, if there's a certain amount of dry forest or a certain amount of mesic forest, then you could try to incorporate both like species that are found in those biomes and see which one like takes better. I think with this analysis, it's still very like in the works, and it's not so much like, oh, this is that solution. It's more of like a suggestion and just hoping people open their eyes to the different possibilities for a place, rather than being like, oh, well, the place is not going to be a mesic forest anymore, so like we're just going to not do anything. I think it's helping people be like, oh, well, it could become a dry forest, which is still better than, like I said, invasive grass. If that helps.
All right, well, let's move on to our last talk, also a terrestrial setting. Riley, if you want to come on up. So Riley Larson, who is also a rising senior and our pilot Payak Surf Fellow, is an environmental resources major at the University of Alaska Southeast in Juneau. And so she's on exchange from Alaska, which is very cool. Riley shares her perspective that in the midst of the rapid changes happening to our planet, it is important to understand the cycles that have been in play for billions of years and those that we have brought on as humans. Climate science helps us to make informed decisions to mitigate our impacts and maintain and restore the natural abundance relied on by all life on Earth. Great statement. So why don't you go ahead and finish us off, Riley? Put that to you. Your shirt somewhere.
All right. So during my time here, I had the honor of working at Napohaku o Hawaiʻi Island, which is how do you?
Is it not? Press the right arrow. Should be.
Oh, okay.
Yeah, there you go.
So it translates to boulders of Hawaiʻi, and it's located right on the edge of Kawainui Marsh over in Kailua. And it's a really important cultural site. That's where Kawainui Fish Pond was, which was like the biggest fish pond on the island and fed about half of Oahu at one point. The Hawaiian legend says that Hawaiʻi was the Amakua, or goddess of the fish pond. And she often took the form of a mo'o, a lizard, or a beautiful woman sunning herself on the rocks. And she brought an abundance of fish to the pond and ensured that those in the ahapua'a shared the wealth, but punished those who were greedy. So just kind of maintain balance. And as you can see in the far picture of the rocks, that's the lizard, but it's now like super overgrown and you can't see it, unfortunately. But yeah, so basically they are restoring this into a Hawaiian dryland forest. It's been riddled with invasive species, so that's where Ahahui Malama i Kalokahi comes into play, and they are like the main stewards of the area. They are a non-profit organization whose mission is to practice, promote, and perpetuate a modern Hawaiian conservation ethic. They do this by working to preserve the native ecosystems and Hawaiian traditions through education, active stewardship, and research. And they've been caretaking this area since the 90s. Their biggest challenge is really maintaining all the invasive species because they don't use any like chemical fertilizers, herbicides, pesticides. So it just takes a lot of manual labor to tear out all the plants. As you can see in the far photos, that was just a before and after of like my time here and all the clearing that we did with kupu and other volunteers. And they've had that cleared out so many times before and it just comes back. So it really takes consistent work out there to make this happen. Most of the support comes from Malama Aina programs at nearby schools Le Jardin and Kalaheo High School, and then some from Kupu and the community as well. So what I wanted to look at out there was how do soil moisture and nutrients impact plant growth, and how could we improve plant growth at Napohaku and kind of advance their efforts to turn it into a native forest. My notes are not working. So some of the goals were to analyze soil moisture and nutrient content to see how this could relate to plant success. Experiment with different soil amendments that could help aid in plant growth and buffer any limiting factors, and then to develop a plan for nutrient analysis and soil amendment implementation that Ahahui could use in the future. They never did have soil testing done there. I believe people came in and took samples and they were going to do it, but then the lab shut down shortly after, so they never got those results. So yeah. So for our test to look at different amendments, we chose uwala, which is culturally significant. It's been a staple in the Hawaiian diet for a very long time. It's really fast growing, especially in comparison to kalo, which takes about like 9 months, I think. And then uwala is more like a couple months, so it produces really quickly. And it's often used in replacement for that in times of needing food quickly. This variety came from a research group at UH with Dr. Kantar, and they're doing a study on a bunch of different varieties, as you can see, that produce as well as the Okinawa potato. It's still kind of up in the air, like they aren't sure exactly what the tubers are like. They're still waiting on them. But we got HM26, and I couldn't really find any more info about that. Yeah. So some of the soil amendments we worked on were vermicompost, thermal compost, green waste, and biochar, which with the biochar, we didn't actually get to it in time just because like the processing and cuttings, it took a while, but they will get into the ground before I leave. We have it all prepped and ready. And then the vermicompost and thermal compost came from a lady named Mindy Jack, who is now working over at UH at the Magoon Station and set up her own processing area over there. And I was really interested in the vermicompost because she supplies it to a lot of farmers and they like swear by it. It's known as like gold. And then she also has thermal compost, and all of it is sourced from food waste from local schools. And then green waste, we just took the leaf litter of all the invasives that we've been pulling to look at that. And then with the biochar, I don't know if you guys have heard about it, but it's basically like charcoal, but it's burned in the absence of oxygen. So its structure is really good for binding to like nutrients and moisture, and it holds on to it for like thousands of years. It's an ancient Aztec soil amendment method. Yeah, and they're really interested in implementing that out there. So because we're looking at so many different things with this, there's a lot of different hypotheses. So one being soil moisture is a limiting factor in some areas on site. Soil nutrient availability will be adequate overall, but higher when amendments are added. And then Uala will grow more productively in soil with amendments than in soil without. And then kind of a guesstimate from low to high growth rates would be control, so nothing, and then green waste, and then hot compost, and then vermicompost with the highest success. And I kind of guessed on this based on like how processed and broken down they are, like vermicompost going through worms and everything. It seems like it would be very like nutritionally available for the plants as compared to hot compost that's still kind of like bigger chunks, not fully broken down. And then green waste, fresh litter that hasn't really broken down yet. So for our method, we chose a Latin square design. It is on a downhill slope and then it's partially shaded. So we chose that to account for like all the different factors, the amendments, the sunlight, and the slope. And then we did a 40-60 amendment to soil ratio, which I found in a paper by Bluen et al. That was looking at vermicompost and different ratios, like what was most successful. And they found that like the golden area was between like 30 and 40% amendment to soil. And then we tracked this based on the number of mature leaves and shoot length. I would go in about once a week and measure that, and water a few times a week. And there's a key over there of what each of the plots were. And then unfortunately, all the 5s are just like empty right now. So for soil sampling, we got samples from a bunch of different areas. So the Uala planting area, which is down in the corner, which is going to be a kukui grove at one point. That's why I chose to look at that area. And then we looked at Uala soil mixed with the thermal compost, mixed with the vermicompost, and then with the green waste. And then there's an acacia koa area. It's the middle top, and it's a very sad looking tree. I think they planted like 5 to 10 and only a few survived. 20 or 30? Yeah, and there's like, yeah, and they're not very happy. And then the ohia area, which there's no ohia there. I think they planted also like 30 of those and none of them survived. So I wanted to look at why that might have been, like what limiting factors were in play. And then under the tree canopy, they want to do a dryland kalo patch. So I chose that as well to look at. So for soil moisture, we looked at the gravimetric basis and then also the volumetric basis. Volumetric was based on this volumetric water spectrometer that basically you just stick it in the ground and then it gives you a % reading of moisture. And then we wanted to test it in the lab as well to like see how it averages, how accurate it might be. So we found bulk density by doing kind of a trash bag experiment. We would dig out a hole and then measure the weight of that soil, put a trash bag in, see how much water filled it, and then we could translate it and calculate it into density. And then we recreated that in those little cylinders with mesh bottoms. And then we could fully saturate it and see what the field capacity is, how much water it could hold, and then also how much soil is actually in there when it's all the way dried down. And then for the gravimetric basis, we just took like 20-gram samples of soil and then put them in the oven and measured them before and after to see how much weight the water was. So for nutrients, we looked at nitrogen and potassium, which are both really important for plant growth. And we started that by doing an anaerobic incubation where we saturated the soil with a little bit more water than like the max that it can hold, so that the nutrients would bind in the water, and then ran it through a what's it called? I'm blanking. Centrifuge. Yes, to separate the solution from the soil. And then we used ion-specific electrodes, which are like 2 little electrodes that you put into the solution and it reads parts per million and the voltage. And then we also wanted to look at a home test kit that they could use at Oʻahu in the future, so they don't need to come into the lab and do all this complicated stuff. And it was super simple. You just mix soil with water, let it settle, and then put some water into those little reader tubes and then add a tablet, and then the color changes over time. So for the Uala planting, we found that thermal compost was actually the most productive amendment of all of them, which was surprising. And then green waste and then vermicompost was not as successful, both with leaf growth and shoot growth rates. And then, so pigs took out like half of my plants like a couple weeks in. So there was 2 separate plantings. So some of the plants were in the ground for like almost 2 months, and then the second batch was only 27 days. So I took the data from day 1, day 14, and day 27 from all of them. So it is a little bit biased just because like climate and moon phases were different. But yeah, it had a trend overall. That makes sense. For soil moisture, we found that with the gravimetric basis that Ohia and Kalo were relatively low in moisture, which would make sense why they, or Koa and Ohia, which would make sense why they weren't as successful. And then vermicompost and thermal compost had like an extremely high rate of water. And then compared to the volumetric basis, the numbers are kind of all over the place, but we found that at the bottom of the hill, it was relatively more moist than at the top of the hill, the water running down. For soil nutrients, our potassium electrode was not working, so we had to kind of scrap that data. But the nitrogen was. So we had to calibrate the nitrogen electrodes before we tested the solutions, and that's what that top curve is, based on parts per million and the megavolts, millivolts. And then, so when we actually did the electrode test, the parts per million were way off, but the megavolts were not. So we kind of calculated the parts per million based on that graph that we had. And then the bottom one, breaking that down. So basically the lower the number, so like the ones in the negatives have the highest concentration of nitrogen, which are in all the amendments. Lowest being in some areas around the Uwala. And then with the rapid test, we found highest nitrogen as well in the amendments. And then potassium was kind of all over the place, and we didn't really find trends with it. That was a more challenging part. And then, so with the numbers on the side, there 0 through 4, 1 was rated deficient on the rapid test, and then 4 was like surplus. So back to the hypotheses, most of them added up. Soil moisture is a limiting factor in some areas. Soil nutrient availability will be adequate overall, but higher with amendments added. We found what Uwala grows most productively with, and it was a completely different order than what I thought it was. And then rapid tests could totally be used for future. But it seems that overall, like the nutrient values across all the sites were pretty stable, and it was the amendments that really changed them, and we saw that they had more productive growth rates. So with thermal compost being the most productive, they could totally set up like a compost area on site and collaborate with the local schools and do their food waste similarly to Mindy. And then the green waste is super easy and accessible with all of the invasives that they tear out. Thank you to everybody. This was such an awesome experience. It was great to be able to work like in the lab on the scientific side, and then also out at Oʻahu getting like the indigenous agroforestry side and kind of finding like the middle between that. Yeah, thank you guys. Do
we have any questions? Brad.
I'll admit this is for my own personal gardening interest.
Yeah.
This question, with the green waste compost, you mentioned a lot of that's made up of invasives and deep mold and everything. Do you have to worry about further spreading invasives using that material?
I was wondering about that too.
Can you mitigate for that?
I think as long as the roots are not like fully intact still and like replanted when you do that, and it's just like the leaf litter, I guess seeds could be a thing too, but then you just have to do some extra weeding. Yeah.
Were you able to look or tell if there's any local fauna that affected your results, like quail or deer or boar?
Definitely boar and mongoose. It could have been the boars or the mongoose that tore out half of my plants. Not sure, but we got the cages up and got rid of that problem.
Another question is the biochar. I'm interested in that. What did you use for the biochar? How did you prepare it?
So it was koa haole, and I went to a demonstration out at the Waimanalo station. There's a grad student out there that's doing a big project on it, and yeah, it's all koa haole, and then you basically get like a huge drum and then a smaller drum inside, and then so the smaller drum is what all the koa haole goes inside, and then you stuff it with like smaller branches and sticks to burn around it, and then you put a good lid on it and you burn it from the outside in and use like as little oxygen, try to block it off as much as possible and just wait for it to burn. And it's actually a really clean burn, surprisingly. There was no like dark smoke at all because all the carbon gets like solidified in this goo char stuff that leaks out of the bottom in a solid form.
Yeah. We actually have, PICASC has supported a graduate student in Guam who was doing a biochar project, and he was working on areas that had been cleared by wildfire, and trying to replant and enhance the replantings with biochar. And they were doing great. And then another fire came through and burned them all down. It was very sad.
I will add too, you have to charge it with something. So they commonly do like fish emulsion, which is what we used, or like compost tea, because if not, like it wants to suck all the nutrients out of the whatever it's in. So if you just put it directly in the soil, it could worsen plant growth because it's taking all of its resources. So yeah.
Interesting. I think there was one question online. Do you want to unmute yourself and ask? Was it Brev? Is that the right name? Nope, I guess not anymore. Okay, well, if we don't have any more questions, that concludes our symposium for this year. And except for 2 students who have 1 more week to go, our surfers have finished their summers. And I'm thoroughly impressed with all of you guys. I think this went really well. So I hope you all enjoyed yourselves, and you will keep in touch because we would love to see what happens to you. You all have a lot of promise. And I think, oh, Brad, you want to say something? Oh, just so you all have lots of promise, and we want to encourage you in your futures. So please do keep in touch and let us know if we can help in any way. Brad.
Thanks, Rachel. Yeah, I just want to echo everything you said. Great presentations, great science. It's really so cool to see how well those presentations were all done. I also want to thank our mentors too for
yes.
I know it's a lot of effort to work with students through the summer. I hope you had a good experience too. I hope you'll return. And along with that, I hope you'll kind of spread the word with your fellow researchers. But mostly I wanted to thank Rachel for all her work. This is the it's really a year-round effort to make this happen with the planning early in the year, and then recruitment, and then the matchmaking process, getting the students with the professors, and then running the program. So yeah, thank you very much, Rachel. All the great.
Yeah, and to add on to that actually is we do have the written requirement, and I hope you all got that email and you know how to do that. And part of that is giving us feedback. So we're always looking to try and make this better. So please do give us any thoughts of what went well, what didn't go so well, and how you think that we might be able to do better on this. So thank you all for coming, others who aren't students and mentors. We really appreciate your support in all of what we do and in keeping these students going. So thanks very much, everybody. And those online.